Well, good afternoon, everybody, and well, good afternoon from Boston, Massachusetts. Good evening or good morning, depending on your time zone. It's great to have you with us today. My name is Frank Slack. I'm the director of the Harvard Medical School Initiative for RNA Medicine, one of the founding institutes in the RNA Collaborative, and we're very proud of how it's grown to be such a worldwide group of great RNA centers. So today we have two of our outstanding junior faculty to present to you. We're going to hear from Dr. Ryan Flynn and Dr. Silvi Rouskin. I'm going to moderate the session. Please will you type your questions into the Q&A, and I will have an opportunity to ask them your questions at the end of the talk. So we're going to start with Dr. Flynn. So Ryan Flynn was an undergraduate at MIT. In fact, started young in the RNA field. He worked in Phil Sharp's lab on non-coding RNAs. And then he moved to Stanford, where he did his MD and his PhD, and continued his work in RNA, working with Howard Chang to develop methods to study RNA-protein interactions. And from this, he won the Weintraub Graduate Student Award, which is for an outstanding graduate thesis. And then as a postdoc, he changed fields to learn about chemistry and glycobiology with Carolyn Bertozzi at Stanford, who just got the Nobel Prize for click chemistries, and was awarded a Damon Runyon Cancer Research Postdoctoral Fellowship. And then in 2021, Ryan opened his own lab at Boston Children's Hospital in the stem cell program and in the Department of Stem Cell and Regenerative Biology at Harvard University. His lab is currently focused on advancing methods and mechanisms surrounding the Glyco RNA molecule, which operates at the interface of RNA biology and the circuitry pathway. So join me in welcoming Dr. Flynn. Take it away, Ryan.
Awesome. Thanks, Frank. Hopefully everyone can hear me. I will get sharing. It's great to be part of this seminar today, and excited to tell people a little bit about the story behind Glyco RNA, how we were thinking about this problem when it started, and a little bit of where we're going now, what we've been realizing and trying to figure out as we've advanced some of the thinking around this. And so, as Frank mentioned, I was sort of trained as an RNA biochemist, and so the questions and the thinking that I have is often biased to imagine this sort of sentence here, which is that RNA really gives life and is central to a lot of biochemical processes. But what I was realizing was that actually where the RNA is very important. So you can imagine that, if you are at home and you're in the kitchen doing something versus in the living room, you might be doing something very different. The activity you would do is potentially distinct, but you're the same person. So the same thing is true for biopolymers and RNA included. And so when we think about where RNA is, it can inform what it's doing. And so we, of course, generate the RNAs from our genome and the nucleus. So there's nuclear RNAs. RNAs are transported into the cytosol for translation and metabolism and regulation. There's a lot of RNA biology that occurs on membranes. An obvious example is translation on the ER. RNAs are sequestered there. Regulation like splicing can happen in response to protein stress. So there's a lot of 2D RNA biology that can occur that's quite interesting. There's also a sort of a growing body of literature around vesicle secretion and encapsulation for transport and communication across cells. But I think when I was thinking about this, one of the critical pieces is that this is actually topologically the same as cytosolic RNA or nuclear RNA. So these vesicles, when they form, capture pieces of the cytosol. So what that left me thinking about was the other spaces in the cell. So the luminal spaces of the ER and the Golgi and the endosome. And these spaces are usually sort of privileged for the purpose of glycosylation and transport and secretion. And so what I was curious about was, is there RNA in these spaces, which led me to the question of actually, how do you test topology, as a readout? Like, how can you figure out if RNAs topologically cross the membrane and get into these luminal spaces? And one of the things, the ways we were thinking about this was, again, sort of the function of these organelles is often to glycosylate and fold proteins, glycosylate lipids, and traffic them. So glycans themselves, if you forget about their actual function, which is a topic of many lectures in length, you can imagine glycans actually just report on if something had ever been brought into the lumen of the ER or Golgi. And so one of the things we're thinking about is if you take a lipid and you put it into the lumen, you get it glycosylated. And if you take a protein, you put it in the lumen, you can get it glycosylated. So what if an RNA got into the lumen? We wondered whether or not it would simply get glycosylated. And I think the benefit or the advantage that I had was that I didn't think about why or how this could happen when we started off. I just wondered if the outcome was possible and wanted to just think about using glycans as this sort of dumb reporter of being luminally located or having luminal topology. And the way we thought about doing this was using chemistry, right? Using the power and specificity of chemistry. And there's this concept that many will be familiar with, which is the metabolic reporter strategy, where you can synthesize a probe, for example, like 5-ethynyluridine or L-azidohomoalanine, feed cells these probes, and they get incorporated into biopolymers that they're specific for, either RNA or proteins. And the critical feature here, of course, is that there are these handles, these chemical handles that are selective for derivatization after lysis or on cells that are specific for the probe you've added. And now 22 years ago, the question was really, can you address this for glycans? And it turns out that my advisor, Carolyn Bertozzi, was very irritated that didn't exist for glycans and went on to invent a series of concepts and chemical tools that allowed us to do this. And so they derived a series of substituted sugars. And in this case, they were looking at sialic acid. So they would take precursors of sialic acid, derivatize them in some way, add them to cells, and they optimized it such that these could get incorporated into sialic acid containing glycans. And one of the first strategies that used this bioorthogonal concept, that invented this bioorthogonal concept, was this phosphine reagent that could click onto azido sugar. So in this case, the azide was that handle that got into the sialic acid, and you could later read out the sialic acids with the phosphine chemistry through some click of a fluorophore or biotin. As Frank mentioned, now 22 years later, and sort of an enormous elaboration of these ideas through Carolyn's work and others, led, of course, to the really exciting announcement a few weeks ago of the Nobel Prize here. But the reason why I'm spending a couple of minutes on this is because, again, this idea, this concept was invented in 99, 2000, and used this sialic acid reporter called MAN-NAS. And it turns out that's exactly what we used when I was in her lab a few years ago when we were thinking about this glyco RNA concept. And so what we did was the exact same experiment they did, which was just taking cells, adding this azido sugar. But the thing that we ended up doing differently was, rather than throwing away the RNA and only looking at the lipids or proteins, we actually looked at only the RNA. And the idea was that if you could extract the RNA, use this bioorthogonal and click chemistry to label the glycan reporter, you might be able to find a conjugate between the MAN-NAS, which had been well established to be incorporated into sialic acids, and cellular RNAs. So it was an otherwise sort of simple idea, but it didn't, of course, fit the way the biology should work with the topological sort of restrictions of RNA and these membranes. So, what does this actually look like? Well, this is just one of the many examples that we had where if you extract RNA from cells that have been treated with MAN-NAS, you can show that we got RNA. So if you do plus and minus RNase, you can destroy the RNA in these lanes and pairs. And if you further transfer this to a membrane and image for that MAN-NAS reporter, which again becomes sialic acid, you can develop these types of blots where in the very high molecular weight region, you can see this blob signal where in the HeLa cells, it's a little bit weaker, and in the ES cells, it's very strong. And this is very exciting. And we did a bunch of characterization to basically define the fact that these were these were really RNA templates, and that this was likely on small RNA templates, so small non-coding RNAs. And to sort of further refine this insight, we ended up sequencing these RNAs. And what we ended up finding was a few things, and many of them were sort of surprising. And so when you look at this plot here on the right-hand side, I'm showing you enrichment values. So the x-axis is the enrichment for the ES cells, and the y-axis is the enrichment for the HeLa cells. And what you can appreciate sort of at a high level is that there's a diagonal line, which means that the RNAs that were enriched in either cell line were very similarly enriched in both. So if you were a glycoRNA in HeLa cells, you're also the same fractional glycoRNA in ES cells. And what we noticed was that there was a select group of small RNAs. So SNO RNAs, yRNAs, uRNAs, and in a different plot I'm not showing, we saw tRNAs. So we found that all of the families of small RNAs were enriched, but not every one of them were. And this was also not dependent on the abundance. So this was not simply that the SNORD36 was the most abundant RNA. There was some other selection mechanism going on to pick the RNAs that were being labeled with MANAs. And so this was telling us that these small RNAs somehow were picked out and selected for modification. But one of the questions is, what glycans? So the glycans I mentioned have these roles, these regulatory roles, but we wanted to know what types of glycans were actually being put on the RNA so we could think about what implications and what functions these could possibly have. In work for just time I won't get into, we had narrowed it down to see that this MAN-NAS signal was generated likely by N glycans. And we could do a few experiments, which I'll show here. First, for example, inhibiting OST, which is the major N-glycosyl-transferase, which is located in the ER. And sort of across a concentration gradient, we could get destruction of the glycoRNA signal. As you can see, the last lane has basically no signal after inhibiting the core N-glycosylation machinery. If we look later in the pathway, for example, with kifinesin and inhibiting a minosidase, this is going to actually both impact the final accumulation of a glycan, but also the configuration. So kifinesin will change the glycoform that are appended to glycans or glycoconjugates. And so what we noticed with kifinesin was very interesting, because inhibiting this enzyme both reduced the signal, but also changed the molecular weight. And so what I didn't mention was that the molecular weight here is a little confusing because it's so highly up running, but the RNAs are very small. And so one of the things that we were getting insights here into was the fact that likely the glycoform, the charge to mass ratio and the structure of the glycans are really impacting this migration, where they're shifting up very high in the gel, these small RNA templates. And we repeated this and sort of extended this result with a different inhibitor called swansonine, which showed us a similar effect that, again, manipulating the glycoform can really have an impact on the molecular weight and the migration and intensity of the glycoRNAs. I think the kind of critical thing that we learned about these glycoRNAs is in the next concept, which is really, where are they? I mentioned that where RNAs are, where biopolymers go, can impact their function. And if these were inside of the cell, they might be doing one thing, but if they're on the surface of a cell, they might be doing another thing. And so we thought if we could swap this experiment a little bit, and we could, rather than homogenizing the cell and losing all of this topological information, if we could first shear off all of the sialic acids from the cell surface, keeping the membrane intact and thus topology intact, we could assess if the sialic acids on the glycoRNAs were on the surface of the cell or if they were on the inside of the cell. And then we could do the processing and make our gels and do our imaging. And what we found actually was that the glycoRNA sialic acids were highly labile to treating living cells with a sialidase, which we inferred meant that most of the glycoRNAs are actually on the surface of a cell while it's alive. And this was very reproducible. It happened across multiple types of cells, whether they were adherent or suspension cells, which was telling us actually that the vast majority of these glycoRNA molecules are actually presented on the external surface of a cell. And this adds a lot of complexity and a lot of biological questions, which I can get into either at the end or in questions and answers. But we wanted to, of course, think through this and try to validate it with an orthogonal approach. And so if it was true that this was sort of the configuration, we could build a model that looks like this, where you have an arbitrary surface of a cell with an RNA and a glycan sort of sticking out. And we thought that maybe the glycan itself would be accessible for binding. And we could use reagents that were selective for binding glycans to stain cells with and assess their RNase activity or their RNase binding properties. And what we found using these Siglec receptor reagents was that actually 2 of about 12 of these, and this includes Siglec 11 here and Siglec 14, were both able to bind cells under normal conditions, but they were sensitive to RNase treatment, which suggested this sort of configuration here, at least in part, is correct, where some of the ligands for Siglec 11 or Siglec 14 are glycans that are mediated by an RNA intermediate, because if you cut them with an RNase, these Siglecs fall off. And so this led us to sort of the summary model here, where not only do we think there are glycoproteins and glycolipids, but we think there are also these glycoRNA molecules that are presented on the surface of cells. They're formed with the template of these small non-coding RNAs, U and SNO and Y and T RNAs. They're composed of these highly sialylated and potentially fucosylated structures with N-glycoforms, and there's a lot more to be done on a structural perspective to sort of really carefully define what's actually going on. Something I didn't mention, but this is widely seen. So we see this on cancer cells and primary cells. We see this in vivo, in vitro. We've seen this across different animal models. Wherever we can find a sialoglycan, we can find these sialoglycoRNAs. And of particular interest for us has been this surface localization. And so thinking a lot about how this could actually contribute to the biology of cells between cells in a tissue, cells in the immune system. A lot of questions can be asked about how these could participate in either regulatory or structural roles. But what we've been thinking about since we published the work are a series of other sort of like technical questions that are, I think, relevant both to glycoRNAs, but also to general RNA biology, because what we realize is that, of course, this is a lot of molecular and chemical manipulation of the RNA that we need to be like highly reproducible and highly robust. And so some of the key questions we've been asking ourselves, which seem sort of very basic, but again, I think they're actually very important. So first of all, is our probe actually labeling all the glycoRNAs? So we used MAN-NAS, but not every glycan may have sialic acid. So are we missing something? When we take cells out, we assume, or we might assume that you're actually extracting all of the RNA or glycoRNAs, but is that true? We also wanted to know in the processing steps, are we losing glycoRNAs? So we actually go through 3 rounds of like cleanups and enzymatic processings and labelings to get from cells into those blots. And that could be a lot. Are we sort of adding variability that we don't understand? And finally, that just even that blotting assay I described, are we losing anything? Are we really seeing all of the material that we've labeled and extracted out of the cells? And the answer to all of these is no, or like 3 is yes. But I'll go through them briefly. So, in terms of are we seeing everything? Again, the answer is probably no. So one thing that we've begun to look at is using other metabolic reporters. And so the first one that I was describing is called peracetylated MAN-NAS or AC4 MAN-NAS, but there's peracetylated GlycNAs and GalNAs and many others. So we did a pilot experiment where we took these other sugars and labeled the cells and extracted the RNA and produced one of these glycoRNA plots. And you can kind of see that there's a bunch of stuff going on. So some don't label. For example, these are HeLa cells. Maybe GlycNAs doesn't get into HeLa cells very well. GalNAs does. It has a similar but different profile than the MAN-NAS. There's higher and lower molecular weight. There's some at different intensities. I think we think this is sort of really just scratching the surface of what are all of the glycoforms, and potentially glycotemplates on the RNA side, and really looking deeper into these other reporters is going to be, I think, very important and interesting. And again, the other sort of, once you have these probes, is now you have to get the stuff out of the cell. And the question is, what's the best way to extract the RNA to actually see the glycoRNAs? And it turns out that we had been using a really inefficient strategy. So when we were doing all of the work previously, we had been triazole extracting the cells, purifying them through various means. And if you don't look at this right-hand side and you produce a larger exposure on the first 3 lanes, that's the signal we've been seeing previously on all the other slides. But what we noticed was that by switching the reagent, the lysis reagent and extraction reagent, we could almost by an order of magnitude increase the amount of glycoRNAs that we can see. And we think this is really important because it means we can use fewer cells. We can do this more rapidly. And also we're going to be seeing a richer population of the material that is in the same cells, right? So we don't want to be losing material at any step. So this was one thing that we were thinking about. But through the processing, we're doing either precipitations or column cleanups. And what we essentially noticed was that we were losing material. And while the columns are, by the manufacturing protocol, very efficient for capturing large RNA, they're quite inefficient at capturing small RNAs. So these first 2 columns here are a comparison of input material versus small RNA that's cleaned up under standard conditions with Zymo columns or other columns. And what you can notice is that every time you do a column, you'd lose 40% of your small RNA. Now, if your large RNAs are your targets, no big deal, but we're looking at small RNAs. And so we have to do 3 columns. So if you take a 40% hit at each column, that's very bad. And that means you're gonna have potentially increased variability, irreproducible results. And so nicely and conveniently, you can actually recover this. You can just switch a little bit of the precipitation conditions and really make these columns quantitative. But this was something that took us like months to figure out because you're like racking your brain, like, why are we getting inconsistent results? Everything is going in carefully. But really a lot of these little steps I think are gonna help us at least, and hopefully others if you're interested in the conditions. The last thing I want to show is again one of these things where we're just using sort of very standard protocols that have been around for maybe decades, but we realized things were inefficient. And so I'll show you a screen, a pH screen of transfer buffer conditions, because we realized sort of that we were not transferring all of the material that we had put into the well onto our membrane. And so it turns out that if you buy the buffer from Thermo Fisher, you'll pay $200 a liter and you will get poor efficiency. So if you start off in the top row, you can run your RNA out. If you do the transfer and then image the gel afterwards, there's still RNA left. And if you look at the last lane compared to the first and fifth lanes, you'll notice that there's more glycoRNAs left in that gel. So when you get complete transfer, either again in the first or fifth lanes, you get way more glycoRNAs on the membrane. And so we've done pH screens and salt screens and things like this, and have found a sort of an optimal buffer condition that allows for what we think is quantitative recovery of total RNA itself, but in addition, glycoRNA. And so I'll stop there to leave a couple minutes for questions and thank people who have been helping with the work. So of course, this work started in Carolyn's lab, and she's just an excellent mentor. I could spend another half hour talking about it, but she's amazing. But since starting the lab here at the Children's Hospital, the team is shown here with pictures. So we're all sort of thinking about these various questions about why and how and where and when about glycoRNA and glycoRNA biology. And thanks to the funding sources so far for the lab. And yeah, I'll pause here. And again, happy to take questions in whatever format's easiest.
Great, thank you very much, Ryan. I'm gonna ask some questions that have come in through the Q&A. We're actually getting a few questions about stoichiometry. So in one question from Ningning Zhang, he or she asks, in cells that have a higher glycan signal, is this caused by more RNAs being glycosylated, or are the RNAs individually more glycosylated?
Yeah, the stoichiometric questions and the flux of these things are relatively difficult for us to still study. I think one thing that we have learned is that when you use metabolic reporters, you're looking at a few things. You're looking at the cell's ability to uptake and convert and use that reporter, as well as the abundance and general accumulation of your conjugate or your biopolymer. This would be true for 4EU or whatever it is, or 5EU. So what we noticed actually was when you go to methods that do not require the metabolic reporters, the true variation in glycoRNA signal is different than what we were seeing with MAD-NAS. Additionally, and this is why I think using other reporters is going to be helpful, or and developing other techniques. Sialic acid is in some way of describing a decorative glycan. It has regulatory activity. Putting it on means something, taking it off means something, but the whole glycan can be there, and the whole RNA or protein or lipid can still be glycosylated. So when we see changes in sialoglycoRNAs, that could be a full glyco change, or it could be a regulatory change on the sialylation of that glycan. And that's something that we don't have vision into yet, because we need tools to study the glycoRNA agnostic from the glycoform. And so we're working on tools like that. But what we're seeing now is sort of just a cross section, I think, and an end point of total amount of sialoglycoRNA versus other conditions.
Great, thanks. We're having a couple of questions also about where these conjugations are happening. So for example, where is the glyco molecule added to the RNA? Is it through the base or through the backbone? And then the other part is, how is the RNA anchored in the membrane?
Yeah, for sure. So, in short, my best guess based on the sequencing data we have is that it's occurring at a G. Because the sequencing data was so specific with RT stops, and it was a specific nucleobase, my guess is that it's a base modification and not a ribose modification. But we're working on, molecular and atomic structures, mass spectrometry, sort of AFM, cryo-EM, all of the acronyms to define that. Until we do that, it's just a guess, basically. But I do think I would bet on it being on a base, likely G. In terms of the membrane anchor, I think we're looking towards other glyco problems. So there are many other highly negatively charged glycopolymers that exist on the cell surface. They have binding proteins. They interact with other cell surface receptors. So I think there's a reasonable sort of paradigm to operate within about where could a negatively charged biopolymer stick to, interact with, move around with on the cell. The short is, I think it's a protein. I think it's likely a protein. I don't think that these are like direct RNA lipid interactions, although that's been published as a thing that can happen. My guess it's a protein RNA interaction through, could be like heparin sulfate binding proteins or chondroitin sulfate binding proteins or things like that, because they're going to have these binding pockets that'll bind negatively charged biopolymers.
Great, yeah. So we had also a bunch of questions. I think you've sort of answered some of it by saying that you think it's being modified at a base. So then the question we're getting from the audience is, what about modified bases? Does that play a role in providing a sort of a molecular cue for where this is going to happen?
Yeah, that's our current hypothesis, that part of the selection criteria for those that are or are not is that precursor modification. It could be a damage event or a modification event, and I think we're working on defining what that is. But I don't think, based on the chemistry, that it's a native base that is derivatized with the glycan. I think there's a primary effect or a primary activity that is like damaging or modifying a base. Then that is the signal for likely uptake into the ER, but in some mechanism or Golgi, and then that can get further elaborated on with the glycan.
All right, and just one last question. I'm going to, there are actually a lot of questions here, so if you could go into the Q&A and sort of type some answers, that'll be great. But there were a lot of questions about how long the RNAs last on the surface. Are you certain that they didn't get there from some extracellular process like dying cells? And are they, can you detect them with some sort of molecular probes like FISH? Can you see them on the surface?
Yeah, we've not had a lot of luck with FISH. short answer, we're still optimizing it. I'm not a imaging expert on the RNA. I think it's very difficult to answer to perfect satisfaction that this doesn't come from some other source, but you can you can digest away everything in a cell, on a cell, and then they will recover in a few hours, and so homogenously. So I think, you know, a cis, like, or a cell autonomous mechanism makes more sense in that context, rather than having constantly enough dead cells to make all of these cells adjacent to them covered. And additionally, you'd have to have a selection mechanism where you're not getting just coating of ribosomal RNA, right? Because these are not the most highly abundant RNAs in the cell. So there'd have to be some selection mechanism whereby an apoptotic cell would release only these RNAs, and these are the only ones that get, you know, adhere to the surface and things like that. But, you know, it's a lot easier to prove a positive, and so we're working on ways to show that something does happen, rather than try to exclude that something doesn't happen, because, you know, you can always come up with a with another model, and so it's a challenge in that way. Yeah.
All right, thank you very much, Ryan. This was great. The audience really loved the talk. There are a lot of questions. If you wouldn't mind going in and typing some answers to give people some satisfaction at the end of your talk, appreciate it.
Yeah, for sure.
So next up we have Dr. Silvi Rouskin, who is currently an assistant professor in the Department of Microbiology at Harvard Medical School. She was actually born in Bulgaria. Her family are not scientists. She tells me that they're musicians, actually rock musicians. She left Bulgaria to work on science in the United States as a teenager, and ended up getting her PhD at the University of California in San Francisco, in Jonathan Weissman's lab, where she pioneered high throughput approaches to measure RNA structure in cells. After that, she started her own lab as a Whitehead Institute fellow, where she established experimental and computational approaches that revealed genome-wide RNA structure heterogeneity of HRV1 with functional impacts on alternative splicing. And then at the onset of the COVID-19 pandemic, her lab determined the genomic structure of SARS-CoV-2 in infected cells and identified alternative structures important for ribosomal frame shifting. She's the recipient of multiple awards, including most recently the 2022 New Investigator Award from the NIH. So welcome, Silvi. Can you share your screen?
Hi, everyone. Yes, let me share my screen. 1 second. There we go. Can you see this?
Yes, looks great.
Amazing. Thank you, Frank, for this wonderful introduction, and thank you for inviting me. I will tell you a little bit about what we've been up to, mostly focusing on viral RNA structures as regulators of gene expression and therapeutic strategies. And in general, my lab's focus is on determining RNA structure, how RNA structure functionally contributes to infection and disease, with the ultimate goal to be able to design therapeutics that directly will target the RNA itself. And some of the lab projects have been a major part of the lab develops method to determine RNA structure in cells, and I'll tell you just a little bit about this. We also have a large focus on understanding how RNA structure and RNA binding proteins regulate viral gene expression, with a very strong recent focus on nidoviruses. And we also have a lot of projects to figure out how RNA modifications affect RNA structure and how this signals and regulates the immune system. And finally, developing RNA therapeutics. And I'm just going to touch back on a few of those topics today, but if you're interested in something that I didn't talk about, feel free to reach out and chat later. So I hope that most of you by now know that RNA structure is important and has a lot of regulatory functions inside the cell. And we really want to know exactly what the structures of all RNAs are and how they're changing in time and space and in different environments. The historically, though, this has been very difficult, and RNA structure has been very difficult to study, and really it's been limited for many years to in vitro studies, which means refolding an RNA inside the test tube, and typically very short RNAs. So here I'm talking about 20 to maybe 100 nucleotides that are somewhat arbitrarily cut out of their full length context and kind of refolded. And then you can look at them with classical methods that have been used to determine structure of proteins, such as X-ray crystallography, NMR, REM, etc. Now, the issue has been that with these techniques that, again, you have to, this environment is very different than the cellular environment, and you are making this assumption that every RNA molecule with the same sequence will actually fold the exact same way, and it will give you the same structure, which is mostly true for proteins. But just to talk a little bit about the energetics, the biophysics of folding, you probably are all familiar with the protein folding energy landscape, and that has been also called the protein folding funnel. And it's a funnel because there is one point of minimum free energy, or there's one most stable conformation that a protein of a given sequence will get trapped into and will fold and wants to fold into. But in contrast, the theoretical, at least theoretical energetic landscape for RNA folding is very different. And it looks more like what I'm going to call an egg carton. And this is because it has multiple wells that are of minimum free energy that are either entirely equivalent or very, very close to each other. But they can represent very different secondary and tertiary structures formed by the exact same sequence. And these structures can give RNA with the same sequence very different properties. And the big issue in the field has been that actually very few long RNAs have been crystallized, and we have really poor prediction ability for long RNA structures, and especially to account for alternative RNA structures. And we're working really hard on this, to establish new tools to address these limitations. One of our favorite approaches is based on chemical probing, and this chemical called dimethyl sulfate, or DMS, that was described more than probably like 40 years ago. We love it because it's very robust. It enters cells really quickly, and it interacts with open and accessible adenine and cytosine bases. So bases that are paired are protected, and they do not interact with DMS. And what we have done more recently is to develop this strategy that for single molecule RNA structure probing that we call DMS-MAP-seq. The idea is very simple. You have an RNA and you can DMS modify multiple open bases on the same molecule. And then we use a special reverse transcription enzyme that simply adds mutations in the cDNA every time that there is a DMS modified nucleotide in the RNA. And now all we have to do is sequence this single DNA molecules. We know if there's a mutation that corresponds to an open base in the parent RNA molecule. And more recently, we developed this algorithm that we call DREAM. It stands for Detecting RNA Folding Samples Using Expectation Maximization Clustering. And the idea here is actually quite simple. Imagine you have an RNA sequence that forms two alternative structures. There will be some bases that are open and exclusive to one structure or the other, and they'll get DMS modified and converted into mutations on DNA molecules. And then when you sequence these DNA molecules, there'll be some co-occurring mutations. And typically what was done in this before our work in the field was you take all the reads and you bin them together, and it gives you some population average signal. Of course, if there are alternative structures, this population average signal doesn't represent any of the alternative. It gets averaged out. But what our algorithm does instead is it looks for these co-occurring mutations, and it separates reads into belonging to distinct clusters or structures, and having distinct DMS signal that we can use now to make accurate secondary structure models. And I'll talk a little bit about that. So using this approach, we did a genome-wide screen and studied HIV-1 alternative splicing in primary cells during infection. And one interesting finding was that we found this very strong 2 alternative structures at this particular splice site called A3 that controls expression of a very important HIV gene called TAT, transcriptional activator. And here what you can see is that simply the 3' splice site is in blue, and you see how in one conformation, and these come from full-length genomic HIV molecules. So they have the exact same sequence, but you see how in one structure, the splice site is really nice, open, and accessible, but in the other structure, the splice site is occluded and paired up. And so we were able to shift this and kind of play with the ratio of these conformations to see how would that affect splicing at this site. And what we found was very kind of cool and interesting, is that there was this really nice anti-correlation with the occluded structure and splicing, meaning that if you push the full genome molecules to this structure, to this conformation, to hide this splice site, you got very little splicing usage and very little production of TAT, indicating that RNA structure is playing a role in these alternative splicing decisions. Another way that we used, this was more of a validation approach, we used the adenine riboswitch, and the nice thing here is the adenine riboswitch is very, very well studied, is beautiful structure. It's been studied a lot in vitro, and the structure is very well defined. And it was more recently realized that actually, even without adding the adenine, there is a small percentage of population of RNA molecules that are already in the on conformation. And when we did a DMS experiment, indeed, we were very quickly able to find 3 major alternative structures, including this on population. And when we added the adenine, the structure shifted entirely towards the on conformation. And this is nice because we can literally, this has been studied a lot, and it's a great positive control. But when we use our methods, we could do this whole experiment within just 1 day, because we can sequence it on our benchtop sequencer, and the actual experimental part is very easy. And 2, we can do this experiment inside cells and see how the cellular environment is affecting these ratios. Okay, so of course, when the pandemic hit, we were very interested and curious to work on the SARS-CoV-2 virus, and we were very fortunate to find collaborators that DMS modified this virus at biosafety level 4 very early during the pandemic, and we were able to determine the genome-wide structure inside cells of this virus that was previously unknown. And in here, I'm just showing you a population average model of just the first about 5,000 nucleotides. This virus is 30,000 nucleotides long. You just see a bunch of stem loops. There are a lot of interesting RNA structures there. Of course, we don't know which one of them matter and how they matter. But one thing that we focused on immediately was seeing this alternative structures at a very particular and interesting element that was already known to be important. It's called frame shift stimulating element, or FSE. And here what I'm showing you is just raw chemical probing data. I'm not expecting you to get too much out of it, except you see there are two major clusters, about 50-50. And this is, again, data from cells. And what you see is that the reactivity of adenine and cytosines, this is the same sequence, is very different on top versus bottom. So some bases are reactive only in one cluster and not the other. So this is just telling you we have two different structures. They're both major conformations. And we then modeling those structures is another point that is difficult, but what we were able to show is that this actually second cluster corresponds to this really interesting and very unusual long distance RNA-RNA interaction. So I'm sparing a lot of slides with experiments here because it's a short talk, but please ask me more if you're interested. But a very talented graduate student in my lab was able to pinpoint the exact nature of those, the exact nucleotides that are interacting with each other. And you can see them here. They're about 1.4 kilobases away from each other. And so we were very curious, this long distance RNA interaction that's very stable, is forming in cells. Is it specific to SARS-CoV-2, or does it happen in other coronaviruses? And this is, I'm going to show you this because I love those experiments. And so we tested a bunch of other coronaviruses in vitro first. So I'm just going to walk you through what this is. This is a viral fragment around 2 kb. This is the frame shift stimulating element, and we're doing this in vitro, but then we've done this in cells also. What you can do is add an antisense oligo to specifically bind and block the FSE, and then ask, okay, does any other base care that you blocked the FSE? Is there any change of the RNA structure far away from where you perturbed the sequence? And I'm just going to show you this results for SARS-2 and SARS-1. So this on the y-axis, I'm plotting the structure correlation, meaning if per position. So the correlation of DMS signal. So if the structure is the same, the DMS signal will be close to 1. And what you can see, this is indeed the case for replicates. On the x-axis, we see position on the SARS-CoV-2 genome. And here I'm plotting all the putative long-distance interactions that one would find if you just did pure computational prediction. There will be many of them. And unfortunately, the computational prediction is not that accurate yet. So we don't know which ones of those, but those are those potential interactions. There's some complementarity going on. But when you do the experiment and you block this oligo here, you add ASO here, in this case, nucleotide about 300. What you see is the structure changes drastically at the place that you added the ASO. That's great. You were expecting this, the ASO bound, but also it actually changed drastically at a very long distance interaction right here. The same thing happens for SARS-2 and SARS-1, and you can pinpoint exactly which bases were interacting with each other. So it's a really nice and quick way to find these long distance interactions. Not all viruses that we tested in this way in vitro were forming these long distance interactions, I should say. You see here, you block this region changes, but nobody else cares about it. So this, in this case, is not forming any long distance interaction. But we were able to identify long distance in a large number of coronaviruses. And it's very interesting that actually going back to the literature, realized coronaviruses have unusually high frame shifting rates, especially during infection. So unlike HIV-1's frame shifting rate is 5%, coronaviruses is 60 to 80%, including viruses that we study in the lab, like OC43 that causes the common cold. And this is showing you data from a very talented tech in my lab, Sarah. And we don't quite yet know what are the roles, the biological roles long distance interactions play, but at least within assays for ribosomal frame shifting, if you allow the formation of this long distance interaction, you get a lot more of frame shifting to happen. So we think that they could be involved in promoting much higher rates of frame shifting for coronaviruses. And more generally, we're now just beginning to uncover this large space of regulation by alternative structures because of applying these new methods. And I told you the story on HIV splicing and a little bit on the story of coronavirus frame shifting. We have unpublished work on how alternative structures control alternative splicing in human genes, including MAPT and telomerase RNA. We now have unpublished data on alternative structures regulating microRNA processing and RNA editing, and we are investigating how that relates to activation of the immune system. And for the last maybe just 2 minutes, or okay, I think 5 minutes, I wanted to briefly tell you about this collaborative work that we did with Anderson R. lab at Berkeley, which is to use antisense oligos as a therapeutic. And the idea here is, imagine you have some important stem loop in, let's say, a viral sequence. You can unwind and destroy the stem loop simply by adding antisense oligo, and if this stem loop was important for the viral fitness, this can be used as a therapeutic. And the idea is very simple, and it actually works just like this in practice, which is exciting. And now I'm going to show you some data. This is again from SARS-CoV-2 genomic region. This is exactly the same plots that we're showing you before. So here you see replicate data. The similarity is very close to 1 because the structures are the same. But when you add an ASO, similar to what I showed you before, you get a large structure change exactly where the ASO bound. So using this kind of chemical probing and clustering approaches, we can actually tell exactly how many viral molecules were bound by the antisense oligo. How specific is it? Is it just binding the viral molecules, or is it binding the viral molecules and a bunch of other cellular RNAs? And what structure exactly is the ASO perturbing? Is it just perturbing the local structure that we meant it to perturb, or is it perturbing some global or larger long distance interaction? And we did a lot of these experiments with different ASOs of SARS-CoV-2. I'm just going to focus on the one that ultimately works therapeutically. So here we're looking at, again, the same kind of data, just plotted slightly differently, looking in vitro at the footprint of the DMS footprint of the ASO over the 5' region of the virus. And what you can see is when you add ASO, you can very nicely see where the ASO bound. You can do this in vitro. You can do the exact same experiments in cells and see where the ASO bound, how much of it bound. You can see, for example, more of it bound 6 hours post-infection versus 12 hours post-infection, and so forth. And and you can make structural models based on this data and find that actually what happens in this particular case is exactly what we meant to happen, meaning there is this first stem loop in coronaviruses that's important for translation, stem loop 1. And when we add the ASO, we just entirely unfold stem loop 1 for them, and we don't see anything else happen. And what's exciting about this, that it also serves as a really good therapeutic for in vivo mouse model for SARS-CoV-2. So if you give the mouse this ASO, you see this great decrease in viral titer in viral protein and in viral protein spike. I particularly like this figure where this is a staining of the mouse lungs for SARS-CoV-2 M protein in kind of brown, and you see if you give it a control oligo, the mouse lung is chock full of virus. But if you give it this ASO, then the mouse lung is clear. So we were very excited about this as a potential therapeutic. And the nice thing about the ASO is that they can be used for any respiratory virus. They're just given by inhalation. And finally, we are working on developing a lot of computational tools to make this chemical probing data analysis much simpler, because the experiments themselves are quite straightforward. It's typically the analysis that is more difficult. So we have, this is collaboration with Joey Yesselman. We have this website called rnadream.org, where you can submit your sequencing files and get a lot of information on the DMS reactivity and structure. So with this, I'd like to thank the people in my lab. These are the current people in my lab here, past people who worked on HIV. I mentioned a lot of the work of Matthew and Amir and Sarah, Lauren Scott, Justin and Mandy is our lab manager that keeps the lab going. We've had some absolutely fantastic collaborators, and none of this would have been possible without funding. And thank you all for listening.
Great, thank you very much, Silvi. That was fantastic. We have a bunch of questions in the Q&A, and I'm going to start just by asking some of them. The first is from an anonymous attendee who says, great talk. They're wondering whether you've tried other less toxic molecules instead of DMS for the structural analysis, and does that change your analysis in any way?
Oh, absolutely. Yes. I personally have tried less and more toxic chemicals, including lead when I was a graduate student. And the issue is that it's really hard. It's been impossible for me personally to find another chemical that has such high signal over noise and is so reproducible and is so it is easy to interpret because it's very specifically targets the Watson and Crick positions. So N1 of A's and N3 of C's. So I have tried quite a few and have stuck with DMS. It's my favorite.
All right, thank you. Aranyak Goswami asks, do you think that there is an RNA version of AlphaFold that will, help the field? And I'm wondering whether Dream, your Dream database is going to be helpful in that regard.
Yes, I'm so glad you asked me this question. I mean, we're not the only ones working on that. There are a few groups working on it. And I think, yes, eventually there will be, I'm hoping sooner than later, an RNA version of AlphaFold. It's more difficult, again, because of these alternative structures and because we have so few ground truth controls, but it's something that we're working on very actively. Yeah.
Okay, and that really brings us to the next question, which is also from an anonymous attendee who says, the two different SARS-CoV-2 conformations that you detected, were they coexisting simultaneously in the same cell, or do they exist in different cells in different states?
Yes. So for the specific conformations that I was talking about on the frame shifting, I think that's what the question is, on the frame shifting element. I can't for sure say that, they didn't come from two different cells, since the way the assay is done, you get, a million cells and you're looking at all the structures that are forming. You're not looking at single cells, right? But those two conformations, specifically those two, they form in the same way in vitro. They're really thermodynamically driven. So they're very similar to what we found for HIV, the Rev responsive element, the RRE, where it's really, the cell seems to have very little control over this because they're so thermodynamically stable. And we see them forming exactly the same way in vitro. So I would personally be surprised if they were forming in different cells. I think they're forming in the same cell. Now, they could be modulated through the cell cycle, such as the exact ratios change a little bit, so you can control frame shifting this way, for example, and that's something that we're looking into.
Yeah, very cool. Rachel Nadira asks, are there any proteins that are known to bind and disrupt the LDIs that you've detected? And would this impact frame shifting rates?
There are no proteins known right now, but yes, there's one protein known called shiftless. Shiftless is a human protein that is trying to interfere with viral. It was discovered more recently with viral frame shifting. So this is one that we're very interested in and kind of following up on. But yeah, they're not known proteins right now. We would love to find some.
Yes, that would be cool. I'm going to just maybe ask one more question. I guess there have been some questions about your ASOs. One is, how do you visualize them to determine their biodistribution? And then, can you use an ASO that spans two distal regions to cause an artificial interaction in cells?
Oh, that's cool. I haven't thought about this. So probably I don't think it will be easy, but it will be super cool. And it seems intuitively, I think it's possible to do this. The difficulty with ASOs is that, when they are in cells, it's hard. We never see them 100% bound. It's always like 10% bound or so. So you can imagine how, it's hard for the ASO to go to the right place to find its target and so forth. But the way was there a question about how we're seeing them binding?
Can you detect them in cells, the ASOs?
Yes, absolutely. Yes. So we can detect them because we are basically probing the structure of the RNA of the virus. And if the ASO is bound there, it just won't interact with DMS. We just get, no signal there. And but we never see it bound in cells like 100% or even 50%. It's usually 10%. But we were working on how can we increase this.
All right, thank you very much. I think we're going to end now because we're a little over time. Let me say a big thank you to both of our speakers. I think the talks were both very exciting. There are a whole bunch of questions in the chat if you want to go through and take a look, Silvi, and just sort of see what other people are excited about. And then
Awesome.
let me just remind everybody that our next RNA Collaborative talk is going to be November 16th, so 2 weeks from now, at 4 o'clock Eastern Standard Time, at the university, hosted by the University of Albany at SUNY. So it'll be great to see everybody there. And once again, thanks to all of our speakers, and thanks to all of you for your attention and your attendance. And we'll see you at the next event. Bye, everybody.